analytical intricacies, regulatory frameworks such as CPCA, the most important, and the forward-looking strategies on how we can systematically mitigate the risk associated with these impurities. So first of all, what are nitrosamines? These are basically organic compounds characterized by the presence of nitroso functional group, which is N single bond, N double bond O. And why they have such importance? Because they have been classified as the probable human carcinogens due to their genotoxic and mutagenic effects. And how they produce those effects is because their ability to alkylate the DNA even at very low concentrations. So the common types are NDMA and NDA. So why this matters now? So it was first raised in 2018, and it prompted a regulatory scrutiny, and also it was initially thought that it's an API related issue. And also it remains an ongoing concern from the API to drug products. So since 2018, nitrosamine impurities have become a significant global concern, leading to widespread regulatory scrutiny across major pharmaceutical markets. Multi-billion unit recalls have been issued since 2018, affecting multiple pharmaceutical companies and generic drug manufacturers. The pharmaceutical industry has been significantly impacted, so the comprehensive risk assessment is required with mitigation strategies. So what is the formation mechanism, how these are formed, and what are the consequences of these impurities? So basically, nitrosamines are formed by through a chemical reaction, which is known as nitrosation reaction, and which involves the interaction of an amine with nitrosating agent. The amine, the secondary amine, tertiary amine, amido secondary amine, or some tertiary, quaternary ammonium salts are also vulnerable to nitrosating agent, and then they can produce the nitrosamine. So the test which is used to assess which kind of compounds can make nitrosamines is called NEP test, which is nitrosation assay procedure. And it's an in vitro method to predict if drug substance or excipient can produce the harmful NDSRIs by reacting with nitrosating agents like nitrites under specific condition, especially under acidic conditions. So the standard standard test for NAP is basically sodium nitrite in water, whereas the modified versions are also available with the organic solvent, for example, tertiary butyl nitrite, to improve the solubility, reactivity, to better assess the risk associated with nitrosamine formation in drug product. So the key components of nitrosamine formation, the key components are secondary or tertiary amine or amide from the secondary amine. These are susceptible to nitrosation. Also, the nitrosating agent, which could provide the nitrosonium ion, and also the acidic pH, which is the most conducive environment for the nitrosamine formation. So the formation relies on three main components: nitrosamine, nitrosatable amine, nitrosating agent, and the conducive conditions such as acidic pH or heat or moisture. When these factors converge, the risk of formation escalates exponentially. And also, so to avoid this to happen, we have to engineer the process of formulation that disrupt this convergence, and if we can eliminate or control one of these factors, we can effectively disrupt the nitrosamine formation pathway. So sources of risk in manufacturing. So there are two kinds of sources. One is the amine sources, and the second is the nitrosating agent sources. So amine sources are basically API degradation. For example, active pharmaceutical ingredients may contain vulnerable amine functional groups that can degrade or react with to form nitrosamines. The second are the solvents. Certain solvents such as dimethyl formamide and methyl pyrrolidone, and dimethyl acetamide, NDMA, may contain or generate the amines during the process. Also, the reagents, amines can be introduced as impurities in reagents or byproducts of chemical reaction during API synthesis. What are the nitrosating agent sources? So raw material impurities like nitrites from the, especially from the, this like control test, such as sodium nitrite may be present in the starting material used in API synthesis. The excipients, common excipients may contain higher level of nitrites or even the trace, which can be converted into the nitrosating agents, and the contaminated water or recovered solvents. They can be one of the sources also. So these are the three cases that I have chosen. Why? Because these are the three different cases. The first one was the famous valsartan case, which was the first time nitrosamine were the issue was raised. So in the valsartan crisis, what happened? The manufacturing process was the NDMA was formed due to the process modification, and the API manufacturer shifted the process, changed the process from tributyl tinazide to sodium azide and sodium nitrite. This seems operationally minor, but it was chemically quite consequential. The interaction of sodium nitrite with DMF containing dimethylamine created a highly risk environment. As we all know that sodium nitrite is highly reactive, and it can be one of the easy sources to the nitrosamine formation. So, because there was no appropriate risk assessment, that's why the formation was easily done. So as we discussed that the sodium azide was and sodium nitrite were used instead of tributyl azide, and DMF was used as a solvent. So the consequence was the DMA converted, the chemical reaction produced the NDMA. So from this case, the key lesson is the process changes require comprehensive impurity risk assessment to identify and control potential nitrosamine formation pathways. So initially, as it was conceived, it was perceived that it's API generated impurity, but ranitidine case defines a new dimension. And in case of ranitidine, it was revealed that the risk profile was different. And the risk profile was associated with intrinsic instability of the molecule. The molecule degraded itself via auto-oxidation to DMA and nitrite under normal, under stress conditions. So this shifted the regulatory narrative dramatically, and nitrosamines and the nitrosamine may generate from the drug substance itself also. So in this one, the key lesson is the stability testing must include stress test conditions and nitrosamine monitoring to account for inherent drug substance degradation pathways. Then the third case was totally bring a new dimension in case of, which is of metformin. So in case of metformin, the root cause was linked to the excipient containing nitrite. The reaction between metformin and nitrite impurities in excipient resulted in the formation of NDMA. Well, this transformed industry understanding also, because it was perceived that excipients are the passive participants, but they can be, by this case, we can learn the lesson that they can be the critical nitrate contributors. So the quality control of excipients are very important. So just to summarize from these key lessons, the first is process changes require comprehensive risk assessment to identify and control potential formation pathways. Also, the stability testing must include stress conditions. And the third is the excipient quality control is very vital to, especially to in the context of nitrate content. So these are the analytical challenges. So in case of nitrosamine impurities, the detection is not easy. It requires the ppb level with signal to noise ratio challenges in analytical detection also. So at ppb ranges, an analytical noise, matrix interference, selectivity challenges can derail the method reliability. And also, if the drug complex matrix interference can also impact the coelution. Also, there can be the co-extraction of structurally similar compounds and chromatographic interference issues. This is why the expectation is very high for advanced technologies and to provide the robust validation frameworks in analytical world. So detecting nitrosamine impurities at ultra trace levels, ppb range in the complex matrix of the drug formulations requires highly sensitive and selective analytical methods with minimal interference. So usually LC-MS-MS and LC-GC-MS are considered as analytical methods gold standards because of their superior selectivity and sensitivity. So these are the four. This is the analytical workflow comparison in which I have tried to cover the established LOQs. So in case of LC-HRMS, the LOQ is 0.001 microgram per ml, whereas in GCHS means head space, mass is 0.002. And for LC-MS-MS, you can see the LOQ is 0.0013, and for GC-MS-MS 0.005. In case of LC-MS-MS, for NDEA, that is diethylamine, the LOQ is 0.0006 micro. So that shows that the LC-MS-MS has the superior sensitivity LOQ, and it can be more sensitive to can produce more better results. So, but this comparison also underscores that there is no single analytical approach to which can be suitable for all scenarios. So the method should be strategically designed, and also the choice should be balanced with the sensitivity and robustness and operational feasibility environment. So method selection should be based on the specific requirements rather than analytical. All of the matrix complexity, expected nitrosamine levels, and regulatory guidelines also must be covered. So the key advantages from these analytical gold methods are ultra trace level sensitivity, high selectivity of nitrosamine compounds, and ability to distinguish between different nitrosamine impurities based on their unique mass spectra. But the analytical challenges still exist. What is this? It's basically because of the complex matrix, the ultra trace level sensitivity is not easy to be attained. So this is one of the analytical challenges, and it has to be improved on day-by-day basis, or the research should be continued. So this is the interactive demo for the immediate release, extended release, liquid suspension, and parenteral solution. Although this is not the universal way, but there's some of the cases that I found in the literature. Most of them prefer LC-MS-MS because of its ability to be more sensitive than GC-MS-MS. GC-MS-MS can also be applied for various compounds, and but the sensitivity-wise, LC-MS-MS is more have the more enhanced sensitivity. So these are the process mitigation strategies, how we can mitigate the nitrosamines. So to break the formation cycle, we must focus on process redesign, solvent substitution, reagent control, and optimized reaction conditions. So first thing, we can the solvent selection, we can eliminate the amine containing solvents such as DMF, NMP, and NMA, diethyl acetamide. So these are, for example, for the compounds which have poor solubility, they can be used. These solvents are used mostly. So if we can avoid these solvents and switch to the non-amine alternatives, that could be that could produce that could block the formation of nitrosamine reaction pathway. Second, so we can if the solvents are inevitable and they have to be used in the formulation, then the separation of the amine containing step with the nitrosating agent could be vital, and that can minimize the simultaneous presence of the reactants and also replace that can make the nitrosamine less happening. So then the alternative reagents like sodium nitrite is very reactive. So if we can change that to with other quenching agents, and the reaction condition should be carefully designed to avoid the reaction to happen. Then quality management system is basically the QA part. So the implementation of supplier qualification programs, establishment of the strict specification for the incoming material is very vital, which can be which can contribute to the mitigation strategies of nitrosamine formation. So this one is the process control mitigation strategies. Now formulation control mitigation strategies. So in case of formulation control, we can control the pH to affect the formation. The nitrosamine formation is favored in the acidic pH, so we can, if we can incorporate sodium carbonate or the basic salts that can provide the basic environment, that it will be more better, and it could make a way for the less conducive nitrosamine formation. Another way, sorry. Another way we can block or control the nitrosamine formation is to introduce the nitrosation blockers and scavengers. So or the modifying excipient selection. So nitrosation blockers and scavengers react with nitrosating agents before they can interact with amines effective. So what happens? They will be neutralized. So they will not be able to react with the amines, and in that case, the nitrosamine cannot be formed. So the two most famous ones are the ascorbic acid, which is a potent antioxidant and react with various nitrosating agent to block the nitrosamine formation. The second one is alpha tocopherol, which is the vitamin E, also an antioxidant. There are various antioxidants that you can use, and they have been mentioned in the FDA guideline with their daily intake limits also. So the mechanism basically is ascorbic acid donates electrons to nitrosating agents and neutralizing them before they can react with amines, and scavengers can compete the amines for nitrosating agents. So this is the introduction to CPCA framework, which is called carcinogenic potency characterization approach. It's a predictive model, and it's developed by the regulatory authorities, and it's the approach which is used to set AI limits, acceptable intake limits, without requiring compound-specific toxicology studies. For example, NDSRIs are very similar to the APIs usually. So the specific toxicity data often unavailable, complicating the establishment of safe, acceptable intake limits. So with this carcinogenic potency characterization approach, we can predict and we can have the acceptable intakes for NDSRIs. So nitrosamines basically are classified into 5 potency classes, with intake limits ranging from low nanogram levels to high thresholds. So how this CPCA framework for NDMA works? So basically, first is the structural analysis, in which the evaluation of chemical structure is used to identify features that influence carcinogenic potency. The second step is assigning scores based on activation and deactivation structural features. And the third step is the placing NDSRIs into one of the 5 predicted potency categories with corresponding air limits. So the sum scores for structural features are, this is also a very vast and very it's individual topic which needs to be contemplated and which needs to be discussed in another way, or maybe in another webinar we can. So in this case, there is alpha hydrogen features, deactivating features, and activating features, and there is a chart in decision tree and chart in the FDA and EMA guidelines, which shows for which functional group what score is needed, and how we can predict the carcinogenic potency classification. So usually these the categories, the range of the categories, like category 1 has the limit of 26.5 nanogram per day according to FDA, and 18 nanogram per day according to EMA. Category 2, which is high potent, is 100 nanogram per day. Category 3 is medium potent, 400 nanogram per day. Category 4 and 5 are similar. The difference is in their the difference for category 4 and 5 can be different. They can be differentiated on the basis of Ames test for their mutagenicity. So this is the chart for the potency characterization, the graphical form, and so the key insight from this one, the CPA-CA framework allows for the science-based determination of safe limits without extensive studies, and categories are based on structural features and metabolic activation potential. So the interactive case, the most important thing is the presence of alpha carbon. So if the alpha carbons are present, it will be it's more vulnerable to nitrosamine formation, so it has to be categorized in a way, and also it has to be calculated on the basis of alpha carbon and the other features. So what could be the implementation roadmap? This is the step-by-step approach which we can a step-by-step approach which we can use for the comprehensive mitigation of nitrosamine impurities, or at least the risk assessment for the nitrosamine impurities. The first is the risk assessment. Second is analytical methods, process control, supplier qualification, monitoring, and compliance. So prevention is more effective than correction. Regulatory expectations continue to evolve, and in the first part, risk assessment, identify nitrosamine precursors. That is very important. The second is how we can map the manufacturing process to mitigate these risks, and the assessment of raw materials. Analytical methods, validation of whichever method, LC-MS is generally preferred, and the implementation of testing protocols. Process control, revision of solvent selection, as we discussed in the previous slide, and also implementation of nitration blockers, control temperature and pH, avoid acidic pH is necessary. Supply qualification is to audit the suppliers, test raw materials, establishment of a strict specification for nitrosamine impurities. Monitoring and compliance is the continuous monitoring, in which the implementation of OOS protocols, OOS procedures, personnel training is very important, and documentation compliance. So the best practices could be integrate nitrosamine testing into quality control, establishment of cross-functional teams, and regularly reviewing the regulations. So these are the key takeaways and future outlook. So nitrosamine risk requires stringent quality control across all manufacturing stages. So this is a systematic. This has to be a systematic risk rather than isolated. So the system is very important. Then analytical excellence, which needs ultra trace level detection and quantification, which is also non-negotiable. The CPCA framework, which could be the facilitator to compliance and ensuring patient safety. Then enhanced analytical capabilities will continue to improve the detection limits and specificity. So there are so many hyphenated techniques nowadays. LC-MS is the highest with the relatively higher specificity and selectivity. Then development of more robust regulatory frameworks for emerging nitrosamine risk. And as we are in the world of AI, so the integration of AI or machine learning tools can improve the risk assessment models. So this is the circle in which the integrated approach could be the process control, the formulation, regulatory frameworks, and the detection. So the takeaway, or the one quote which we can take from this presentation: the comprehensive science-based approach is crucial for ensuring patient safety and maintaining drug supply integrity. And these are the guidelines on nitrosamine impurities. You can see the FDA guidelines, WHO, TGA, the Australian guideline, Health Canada. So recently, TGA published one revision in which there is a revision. They have incorporated more AI daily intakes for the acceptable intakes for the compounds, so we can check the latest guideline. Thank you. So if anyone has some question, they can ask. I'm still.
Thank you so much, Dr. Siddiqui. That was a vital look at the implementation roadmap and the interactive case study. And to our audience, thank you so much for joining PharmaCourse today. If you have further questions, please email us. I will be sending the contact email right now in the chat, or you can visit pharmacourse.com. Have a wonderful afternoon.